Hi everyone, my name is Ved and today I'm going to present to you our work on sequence predictability score to boost classification of protein sequences. So a little bit of background. Currently amino acid sequences corresponding to spike proteins have been successfully used to classify variants. And we noticed that the protein signature for variants of concern and variants of interest are quite distinctive. However, there exist many uncategorized variants which display a high level of heterogeneity. And when such categories are thrown into the mix of a multi-class classifier, everything goes haywire, as we'll show in a bit. So the critical problem is, how can we handle such high level of heterogeneity in the amino acid sequences so that we can develop a robust multi-class classifier model? The data for this project comes from GSAID, and the variant information was determined as per the definition set by the CDC. The overall prediction task is quite simple. We want to be able to create a multi-class classification model that can predict the variants for the sequences. And in order to do that, we start very simple by using a count vectorizer with a balanced random forest. So the count vectorizer just counts how many times a particular amino acid appeared in the sequence. And the balanced random forest is just a variant of the usual random forest, but accounts for the imbalance in the categories. So we use a 5 by 5 nested cross validation, and the rationale is that the nested cross validation allows us to hyperparameter tune as well as get an estimate of the generalization error at the same time. Looking at the confusion matrix, we see that the delta variant is often confused for the others. And when we take a look at the two-dimensional TSNE plot for the count vectors of these two categories, this confusion sort of makes sense because you see these 2 points for these two categories being so mixed with each other. So in order to create a robust multi-class classification model, we need to handle the confusion between delta and others. And we approach this from a sequence to sequence perspective. And the central idea being that if we just use the left-hand side truncated sequence to predict the right-hand side truncated sequence, the kind of errors we would see in that prediction would be very different for, let's say, a tightly clustered category that has a lot of signal and a less tightly packed category which has a lot of noise. And in the categories which are tightly clustered, even if we tinker and tailor with the right hand side sequence, the predictive accuracy for the right hand side will not be impacted as much. However, on the other hand, if we do similar analysis for these more heterogeneous categories like others, we should see a lower predictive accuracy in predicting the right hand side. So let's talk a little bit about our seek-to-seek model architecture. You can see this is the overall network flow for the architecture, which I'll use to explain. The overall architecture has 3 blocks. Let's first look at the amino acid block encoding network. So what we do is we chunk the amino acid into blocks of, let's say, size 20, and the amino acid encoding network just learns a d-dimension vectored representation for each of these unique blocks, and it learns them jointly. Once we have these embeddings in the encoder part of the seek-to-seek, we read in the embeddings temporally in sequence, pass it through an LSTM, and for each time step, we get an output or a vector, which we can then use an attention weighing mechanism to take the weighted average of those vectors to come up with a single vector as shown over here. Once we have this encoded representation of the truncated half, we pass them to our decoder, which uses another LSTM to sort of just process it, and using a softmax operation is able to predict the exact block IDs for the second half. So that's sort of like the very high level explanation for our seek to seek network. And now we will just look at the implementational details of the seek to seek. First, we are only interested in the delta and the others category. So what we do is we take a query sequence, which should either be in delta or in others, and we replace its left segment or the left side truncated sequence by randomly drawing sequences from all the known delta categories or delta sequences in the training set. We then train the seek to seek using these scrambled left side sequences to predict the right side sequences. And under the interchangeability hypothesis, if we notice that the randomly perturbed first segment is coming from delta, and we notice a low prediction accuracy for the right hand side, the intuition is that such a segment probably comes from an other sequence, not a delta category. So that's sort of like the implementational idea. For the model training, you can see all three networks are connected. That means we can train them end to end using categorical cross entropy. We use the Adam optimizer, which is an advancement over usual SGD as it uses an inbuilt momentum, and we use early stopping using a 20% validation set. So in the results, what we see is that if the sequence actually belongs to delta and we replace the left hand segment by randomly drawing from the corpus of delta sequences, the accuracy for the right hand side is 95%. But if the sequence belongs to others and we perturb the left hand side sequence with delta, the accuracy drops to 92%, which kind of tells that this method might be predictive for differentiating between delta and others. That's all I had for this presentation. Thank you for listening to this presentation. And if you have any questions, I would love to hear from you. Thank you and have a nice day.